Cells respond to the environment through signal transduction pathways, which also have a major effect on gene expression. So, down here we have three different types of pathogenic bacteria, and they will respond to changes in population density. So, when they're alone, they're not going to cause much trouble. They're not going to make you sick. They're not even going to release too much as far as toxins go. But as the population increases, they are going to continuously produce these autoinducer, these signaling molecules. And when it gets to a dense enough proportion here, they respond to that change in the environment from these signaling molecules, and we have quorum sensing. And again, it's at this point that they start creating biofilms to protect themselves and start releasing toxins that make you sick. In the case of cholera, you're going to have extreme fluid loss through vomiting and diarrhea. Pseudomonas aeruginosa has a lot to do with sepsis or blood poisoning, if you've heard of that. And you've probably heard of staph infections. So, our sympathetic nervous system is our fight or flight version of our nervous system, and it's going to respond to environmental conditions. If it's cold, if there's a lot of pain, if there's physical stress, your sympathetic nervous system is going to respond, and it connects to your endocrine system as well in the releasing of hormones. Now, what's also unique is that it will also respond to psychological stress. So if you see something scary, you might respond through your fight or flight response, and that includes mobilization of energy stores. So in this process, the sympathetic nervous system will stimulate epinephrine and norepinephrine release from your adrenal glands. And when we look at that, epinephrine release will bind to the receptor protein, which again activates this adenylyl cyclase, this enzyme, which creates the secondary messenger, cyclic AMP. And it's this CAMP that is going to go through a cascade of activating enzymes, which will break down stored glycogen into glucose, which again, this is going to go out and mobilize energy stores and be able to send glucose to cells that are in need of producing a lot of ATP in times of stress. So this idea also another example of how not only cells respond to the environment, but how it affects gene expression and cell response or cell replication. So you have B cells, and they're going to come in contact with an antigen, and activated helper T cells will contact those B cells and send out cytokines. And these cytokines, these messengers, are going to tell the B cell to ultimately go through clonal expansion, make a whole lot of copies of itself, and then those will differentiate into plasma cells, which become factories for cranking out antibodies or memory B cells. So another example here, we have yeast, and haploid yeast will release pheromones, and it's these pheromones that are going to trigger mating amongst the yeast. So these pheromones land, again, we have this complicated signal transduction pathway, and it's ultimately getting involved with the nucleus. And they say there's expression of 200 genes when this happens, and we get this shmoo formation. Some of the genes that are created starts breaking down the cytoskeleton so that through chemotaxis, these two haploid yeast cells can actually move towards each other. They'll digest the cell wall, and you'll have plasma membrane fusion, which results in fertilization. And those two haploid cells become a zygote. They become diploid, which then they can go through meiosis and go back to haploid spores. So again, another example here, when we're talking about signal transduction pathways influencing gene expression and cell function. Plants are going to make ethylene, and ethylene causes plants to ripen. So when ethylene lands, We have a signal transduction pathway that's going to influence gene expression. And you're going to get this ACE, this enzyme created, which ends up breaking down the pectin and softens the fruit. If no ethylene is present, you're not going to have the signal transduction pathway and you will not ripen the fruit. So this idea of gene expression, again, this comes back to this idea of steroid hormones, which are lipids able to go through the lipid bilayer, binding to the receptor protein, and that hormone receptor complex activates genes and ultimately starts transcription. And again, as a review, we have our messenger RNA, which goes to our ribosome. Then we have translation, and we create the functional protein. And again, proteins kind of do a whole lot. But what does it mean to express a gene? When we look at DNA, not the entire sequence of DNA is made up of genes. Genes are recipes for proteins. So, in order to express a gene, not every single cell is making every single protein and doing transcription of every single gene at every single moment. We turn on genes and turn off genes, and that's greatly going to affect the phenotype of an individual or the functioning of that individual as well. So we have to have, again, this idea of receptors, and we can have hormone receptor complexes. When they join up, the receptor side is going to change shape, and then we call that an activator. And the activators are going to land on the enhancer regions. So this is one step in the expression of a gene or the turning on of a gene. In addition to activators landing on enhancer regions, we need transcription factors landing on the promoter region. And it's at this promoter region that RNA polymerase is going to bind. And RNA polymerase is what will actually do the transcription and make the messenger RNA. So without these transcription factors, it's much, much, much more difficult for RNA polymerase to bind to the promoter region and begin transcription. So they hook up, that holds it here as it's moving along, but then ultimately we have to bring the activators over through this DNA bending protein, and then we can kick off transcription, and now we've turned on a gene. But there's a lot of things that have to go into that. We're not just cranking out messenger RNA every single second and cranking out protein for every single gene. So when we start looking at that, an example we can look at is this SRY gene. Again, this gene has to be turned on. When a fetus is developing, the gonads are identical. They are undifferentiated. So male and females begin with the same gonad formation. It's the presence of the SRY gene, which is found on the Y chromosome, which increases the production of this SOX9 gene and creating of the SOX9 protein, which then forms testes instead of ovaries. So as we look at this, again, this SRY protein from the SRY gene goes back to this idea of a transcription factor. And SRY protein is a transcription factor for the SOX9 gene, which then also makes a transcription factor protein, which gets, again, down the rabbit hole. But this idea that we are going to increase the amount of SOX9 gene by the production of the SRY protein transcription factor. So once this gets turned on, we can make a lot more of this. And again, women need this as well, because although they're not going to create it in nearly as much, SOX9 is not found on the Y chromosome, and it's made by women, but it also plays a major role in the formation of cartilage. But in order for testes to form, you have to have a lot more SOX9. So that SRY gene is going to be really important, and it has effects with 6 to 8 weeks after the fetus forms. So again, we have this undifferentiated gonad, and with the SRY gene is going to stimulate the production of Sertoli cells, these ones right here. So that's the Sertoli cells. They're going to help start creating the seminiferous tubules where the sperm are going to drop in. But it also creates anti-Mullerian hormone, and it's this hormone right here that ultimately prevents the creation of fallopian tubes in a uterus. So without this SRY gene, we're going to the follicles are formed, the primary follicles before birth, and the ovary is going to develop. And without this hormone in high amounts, fallopian tubes in uterus will be developed. So again, all of this comes back to this idea that signal transduction is going to affect gene expression, and the gene expression is going to affect the phenotype. So again, if we don't have signal transduction creating the SRY gene affecting the gene expression of the SOX9 gene, we're going to develop into have female characteristics instead of male characteristics. So last example here, we have Hox genes. And Hox genes are homeotic genes. And they have this region of the homeo box. And this, again, this is another gene that codes for transcription factors. And it's this homeo domain that is what is actually attaching to the DNA. So Hox proteins are, again, transcription factor proteins, and they are going to target specific genes. Now, these Hox genes are highly conserved, which means that lots of different things have the exact same genes, even if they're very different phenotypes. However, especially during embryo development, although it happens at different times, the sequence of which these are turned on determines the body arrangement. So if this process is messed up, you can have fruit flies growing legs out of their head. Or if the timing is off, you can get 4 wings instead of 2 wings. And it's from this that sometimes mutations and disorders occur, but also it's how we get differences in phenotype. So again, these Hox genes have to be turned on. You're going to have expression, and if that expression lasts for an extended period of time, you're going to end up with a different phenotype. So during embryonic development of chicken, mice, and snakes, they all have this HOXC8 gene that is responsible for turning on target genes that determine how many thoracic vertebrae they have. So the longer this gene is active or this gene is expressed, the more thoracic vertebrae they're going to have. So as far as a chicken goes, a chicken has 7 thoracic vertebrae. A mouse is going to have 12 because its gene has been expressed for a longer period of time. It's the same gene, it's just expressed differently, whereas a python is going to have hundreds of thoracic vertebrae. And again, all of that comes back to signal transduction affecting gene expression, which then affects the genotype. So again, signal transduction pathways are activated sometimes by the environment, but it is the signal transduction pathways that result in changes in gene expression, which again changes many things about organisms.